also for conservation. So what do you do here? We create something called as barcodes, which are unique identifiers like what we get in the supermarket. So if you go to DMart or if you go to a Reliance store, they have a barcode for each and every uh, instrument or equipment or grocery item that is placed in their center. You just have to scan it, and based on the barcode you can get the information of what it is, what is the weight, what is the price. Okay, so we create unique barcodes for animals, and this method is very fast, very quick, very cost effective. The thing is, you just have to get a single specimen, and you can tell the difference between two species very easily based on this unique method called as DNA barcoding. It can also help you to identify new species. It can help you to differentiate between species. It can help you to even identify new species. Okay, now you can even identify species from soil. You can even identify species from air. Okay, a good study happened from NCCS in Pune, where they collected air samples from air balloons, and they identified the bacteria, okay, that is present in the air. Okay, we can also identify species from water samples. You need not go and catch the specimen, right? So all these samples or all these parts of the body, right, can be used to create a unique identifier called as DNA barcode. And with the help of this, we can identify species, we can delineate new species, and you can monitor changes in aquatic communities or terrestrial communities over a period of time rapidly and very cost effectively. Now, going further with DNA barcoding, for DNA barcoding, you need to catch hold of a species. Okay, you need to have the part of a species. You need to have a hair, or you need to have a leg, which from which you can take out the DNA. Okay, here in eDNA and next gen sequencing, which is called as environmental DNA, what you can do is you can get the idea about the diversity and abundance of the species present in an ecosystem, say aquatic habitat, without even disrupting the ecosystem, without even touching the animal. So you just go there, you take the water sample, you bring it to the lab, and based on the trace amounts of DNA that is present in the water sample or the air sample or the soil sample, and with the help of next gen sequencing, high thorough output sequencing, you can profile thousands of species simultaneously from a single sample. So here in DNA barcoding technique, which is now becoming older, you have to catch hold of a specimen, take out DNA from a single species, and you will be able to identify a single species. Here you can bring 100 ml of water, and from that 100 ml of water, you can get the diversity of species that are present at the given time in the next season, or maybe even in the next season, at a single instance. So eDNA and next gen sequencing have revolutionized biodiversity analysis, have revolutionized the way in which we assess biodiversity. Okay, another benefit of this method is sensitivity. Okay, so we need very trace amount of sample to be detected in the eDNA methodology, and even if it is present in very low amounts in the environment, still we can identify species. Okay, so you go to a particular habitat, and in the habitat DNA will be released via excreta or from the body hair or from the skin, and everything will fall down into the soil or into the aquatic habitat. And these are DNA sequences which are unique to the individual. You can go and collect the sample. You can collect it from water, or you can collect it from sediment, or you can collect it from soil, extract the DNA, amplify it using a PCR machine or thermal cycler, and using a next gen sequencer, sequence it, and based on the information that you get in huge amount, uh, you apply a barcode to it, and you can easily identify most of the times all the species that are present in the given habitat. Okay, so this can help you to identify a single species, but rapidly and cost effectively. This can help you to identify practically all the species that are present in the given habitat at a given instance of time. But this is slightly costlier, and in India it has not been yet developed, but in USA, Canada, and European countries, especially in Canada, they are using this methodology in huge numbers. Okay. Lastly, we move to a very commonly ignored way of documenting diversity, which is called as citizen science. Okay, so what do you mean by citizen science? So, because you are PhD students, because I am a teacher and I am a researcher, we are actively engaged in research. There are people in the society who are also interested in contributing to the research that is happening, especially research which is related to the environment, research which is related to the nature. Okay, so what happens in citizen science is we use children, we take the help of people near in our building, or in our college, or in our society, okay, our relatives or friends, to document biodiversity that is present in a given area. Okay, so for example, if I'm a PhD student and I'm working on birds of say Maharashtra, I may not be able to go to each and every place. Right? Say if I'm present in Ahmednagar College, right? The amount of area that I can cover in a given amount of time is very restricted, right? So I cannot go to Sambhajinagar, I can't go to Pune, or I can't go to Konkan. So what I'll do is I'll take help of the common public, students especially, and they will help me to monitor data and also to collect data which I then can use for my analysis, for my studies, or for conservation efforts. Okay, so there are many agencies in and around outside India which are using citizen science to document biodiversity and also to understand trends in biodiversity and also for conservation efforts. Okay, so one of the most celebrated citizen science activity was done from NCBS and ISC Bangalore, where they looked at butterflies, where they looked at odonates, which is also an ongoing project, and many new species and many new information about distribution of endemic or what do you say, uh, organisms which are elusive, which you normally cannot see. You go to a forest and you're not able to find that organism. Such kind of reports have been done by common public. Okay, and these studies have been published in good journals like Nature, right? So what is the takeaway lesson from here is we can observe animals and plants in the environment directly or indirectly. We can use different methodologies to collect data for diversity, data for abundance. You can use remote sensing methods. You can use advanced techniques such as DNA barcoding, eDNA, and next gen sequencing. And you can also take help of the common public or people who are not actively engaged in research to gather your biodiversity data. Okay, so I'll end with this small example, which we just, the study that we just concluded a few weeks back. So if you look at this particular image, okay, so you can see that there are three different images, and if you look at the all the three specimens, so this is specimen number one, this is specimen number two, and this is specimen number three. If you look at it grossly, you will not be able to understand that these three are three different species. So for a common man or a armature, uh, if you try to identify this species, you will conclude it to be a single species. Okay, but if you look at it in more detail morphologically, you will be then only able to conclude that these are three different species. Okay, so this is specimen number one, specimen number two, and specimen number three. And as I told you, they look morphologically extremely similar. Okay, very minor differences. So what may happen is in the course of your PhD work, it may happen that there are three species in your water body, or there are 3 species in your forest or in your habitat in which you are sampling, but because you are not able to identify it properly because of advanced techniques and methodology not present with you, you will conclude it to be a single species, right? And it may happen that this specimen number 1 may have restricted distribution, specimen number 3 may have very wide distribution, and because you are finding, you have concluded your species to be specimen number 3, and you also count this endemic and restricted distribution species along with this, you will not be able to protect or conserve this species. So wrong identification, right? Because of similar morphology, we call such kind of species cryptic species, can lead to underestimation of biodiversity. Underestimation of biodiversity, right? Underestimation of abundance of the species that are present here. So maybe I'll say this is species number 1 with 3 individuals. Okay, so I'll say the population of this species is very good, but actually they are totally different species. Okay, so moving forward, so as I told you, this is species number 1, this is species number 2, this is species number 3. This is a organism called as large branchiopod crustacean, which is commonly called as fairy shrimps, right? So as I told you, this is species 1, species 2, species 3. So this is a common species found in Maharashtra and India. This is very commonly found in Maharashtra and India, and this was a species that we found only from a single locality, but we were concluding it to be the species B or species number 2, right? And because this species also looks similar, maybe we were also considering this species as specimen number 2. But what we did is, we looked at its morphology, we looked at its morphometry, we did something called as scanning electron microscopy, right? And we also did the DNA barcoding of this particular specimen. Now, with this data in hand, morphology data, morphometry data, ecological data, environmental data, and molecular data, we came to a conclusion that there are 3 species that we find in Maharashtra. Okay, so we're talking here about Maharashtra. So this was the species which I said is commonly found in India and also in Maharashtra. This was the other species which is found in the Western Ghats. That's why the name Streptocephalus siderensis, which is found in the Western Ghats towards the Goa and the Karnataka side. And this was the species that we found, which we were feeling was this particular species. But when we did morphological studies, we came to a conclusion that this might be a different species. But because of molecular data that we generated based on the 16S rRNA gene, we came to a conclusion along with morphological data that we had that this is a totally different species, right? So because of biodiversity methods, advanced biodiversity methods, we could conclude that this is a new species to science and not the original species that we were finding all over Maharashtra, not the species that we were finding in Western Ghats. This is a totally different species restricted to the Western Ghats near the Gujarat area. So what we can conclude is this is a well distributed species where we should or may not concentrate our conservation effort. This is a species found in the Western Ghats, but in many areas this is a species found only from a single locality. So if I am looking for conservation methods, if I have money to conserve these 3 species, I will put an emphasis on this particular species, then on this species, and then on this particular species. So what happens here is, by using advanced biodiversity techniques or taxonomic methods, it can help you to understand differences between species. So this is male and female, differences between species, and you can also understand, or you can even go for a new species to science. So we have just completed writing this paper, we have submitted it to a journal, and maybe next year we may come out with this particular paper where this will be the 6th species from India and the 2nd new species from Maharashtra. Okay, so this is all about the different methods of biodiversity assessment. Okay, thank you for your